Yeah, it's a week from today, Monday, the 20th. So there's no class Monday, the 20th. But you will have a class period, 75 minutes, anytime throughout the day to take the test.
And it's only one attempt, right?
Yes.
All right, thank you.
Oh,
You said Monday the 20th is no class?
that's right.
Okay, I'm gonna mark this down now. If you don't see me, don't mark me absent.
Professor, can you hear me?
Yeah.
Remind me. This is Alex Alice, by the way. What will the quiz 3 like? What's covered on it? Is it everything from last week in terms of the slides? That's it.
Everything since the last quiz. So, let's see. So that will be chemistry for biology, biomolecules, which I'll finish today, and DNA, RNA, and genes, which I will also finish today.
Okay, so it's everything from today and last Monday, right? All the slides from last Monday.
Yes. Yeah, those three things.
I have a quick question too. This
Your
is
name?
Trinity Daniels.
Okay.
You extended the worksheet, correct? You
Yes.
extended the due date?
Uh-huh.
Okay.
Yeah, I
and
did
when is
that
it due now?
because I got out of sync. I assigned a worksheet before I had covered that material in lecture. And so when I realized that, I extended the due date. Some of you have gone ahead and completed the worksheet before I covered the material. And when we go over the worksheet, I will point out that if you did that, I gave you credit even for things you didn't know because you couldn't possibly know them. But that was only one or two questions on the worksheet. Let's see. Yeah, that worksheet is due tomorrow, the 14th tomorrow, and then
Okay, thanks.
we'll discuss it. And you should be able to do it if after class period today, you should have all the answers for that. Then we'll discuss it on Wednesday. I've assigned another worksheet, which isn't due until Tuesday, September 21st. And we haven't covered that material at all yet, so don't try to do it. I should say we haven't covered all of it yet, some of it. But I'd recommend you don't try to do the worksheet until after lecture on Wednesday. Okay.
Professor, this is Steve Stinson.
Uh-huh.
What worksheet are you talking about?
I'm talking about DNA, amino acids, nucleic acids, and proteins. It's the one where you'll need to draw the structure of a peptide bond.
Okay.
And then answer a few questions about the nature of DNA and RNA.
But how are we gonna draw something if it's an online test or quiz?
There
You
are
could draw it on a piece of paper and take a picture of it.
people have offered various different ways of doing it. So whatever works best for you. I know that there are drawing programs that you can use. It's the structure of a peptide bond is fairly simple, so you might be able to do it. In fact, I've done it for myself with just what's available in a word processor.
Professor, just to be clear, you're talking about the worksheet that's due next week on the 21st, right?
That's right.
Okay, thank you.
Right, I would like now to share my screen with you.
Professor,
Yeah,
before you start, because I don't want to interrupt you in the middle of it, I was wondering, I don't remember your policy with attending like Zoom meetings. Does that affect our grades a lot?
no, attendance doesn't matter at all. Participation matters. So you're Elizabeth Martin. So when you speak up in class, I try to make note of that. And I periodically send out my record of who has participated and who has not. And if I don't have it right, then I want you to correct it. Tell me what I've missed so that I have the full record of everyone who's participated in class.
Okay, thank you.
All right, I started this slide set last time, so there's no need to review everything that I've already done. But we're going to continue talking about biomolecules. These are large molecules that are essential for life. And we'll come back to some of these later, but I think we stopped at about this point where we're talking about what is a protein. Protein is a series of amino acids linked together by peptide bonds. The linkages are caused by what's called a dehydration reaction. The reason it's called that is that when it happens, a molecule of water is eliminated. So if you end up with a carboxyl end of one amino acid next to an amino end of another amino acid, you have a hydrogen and a hydroxyl adjacent to each other, and that is H2O, and that is eliminated as a molecule. So you end up with the N of one amino acid joined to a C of another amino acid, and that is a peptide bond. Now, not all NC bonds are peptide bonds. It has to be in this particular configuration. You can probably see that better. Well, this just elaborates on what is a dehydration reaction or a dehydration synthesis, where a molecule of water is removed and two molecules are joined. And that is the opposite of hydrolysis, where you take two molecules that are joined, add a molecule of water, and you end up with hydroxyl on each of the members and end up with two molecules instead of one. All right, this is what will come up on your worksheet, where you're asked to draw a peptide bond. So as I said, every amino acid has an amine group and a carboxyl group, a hydrogen on the central atom, and then what's called an R group. And it's the R group that's variable, and that's what distinguishes each of the 20 amino acids. So you see here on the right, the standard configuration on top, amine, carbon, carboxyl, and then this particular R group, tyrosine, causes the amino acid to be tyrosine. Same thing, but a different R group causes it to be glutamine. So this is what I mean by a peptide bond here on the upper left. We have two amino acids, carboxyl on one end of one, amine on the one end of the other, and a water molecule comes out forming this bond. So this N is now connected to this C through a single bond right here, and that is a peptide bond. Now, here is another C connected to an N. That is not a peptide bond because it's not in the right context. Peptide bond is a CN bond flanked by a carbonyl and an NH. So C double bond O, NH, C and N connected by a bond. That is a peptide bond. And that's what you'll find in every protein. Every amino acid that's joined to any other amino acid is joined by a peptide bond. So if you look at the right here, all these blue balls, each of them is an amino acid, and they're all joined by peptide bonds. And at the bottom, I put this in to sort of make clear what the peptide bond is. The C double bond O, the NH, the bond between the C and the N is a peptide bond.
I have a question.
Yeah,
Is a peptide bond only like, is it only present between carbon and nitrogen?
yes,
Or, okay,
yes. It's only between carbon and nitrogen, and it's only a carbon that's C double bond O and a nitrogen that is NH.
Excuse me, I have a question too.
Uh-huh.
Amos and Rafa. And if we're gonna, like you said, you're gonna draw. So we can draw one of these or we have to create another one.
The worksheet tells you
Oh,
what
okay.
to do.
So, sorry, this is Alicia Carroll. So the peptide bond is going to be always that formula that was in the box, the blue box.
Yes.
Okay.
The-
So the C
the
double
blue-
bond O.
Yeah, this blue box is a peptide bond, and it's the only thing that's a peptide bond.
Okay.
A CN bond without this configuration or without this context is not a peptide bond. And that's illustrated here to the left where you have a CN bond, but it's not in the right context. And so it's not a peptide bond. And the critical thing here is that a non-peptide bond doesn't join two amino acids, whereas a peptide bond does. Here are the 20 amino acids that are found in living things. And each one is distinguished by its side chain, which is referred to as R. The first thing I want you to notice is that all 20 of these have the same configuration. An amine group, a carboxyl group, a CH, and in the case of glycine, the simplest amino acid, the R group, is a hydrogen atom. But then you have alanine, the number 2 item here. Its R group is a methyl, and that makes it alanine. And go on, you can go on and go through all of these. And there's just one other point I want to make, and that is 2 of the amino acids, methionine and cysteine, have sulfur in them. And I emphasize this because it's unusual that an amino acid has sulfur, but they have them for a reason. And that is in order for a protein to fold properly, and it has to be properly folded in order to have activity. We'll talk more about this later, but sulfur atoms are critical in protein folding. So that's why I emphasize it here, along with all the other amino acids that don't have sulfur in them.
Will we have to memorize this?
Say that again?
Will we have to have memorized how many amino acids there are and like all their
No,
structures or no?
no, you don't have to know the structures of all the amino acids. It's easy for you to look them up if you ever need to know the structure. So I don't ask you to memorize them.
Okay, thank you.
Just to compare the functional groups in amino acids with other functional groups that you'll commonly see, I show you this set. So the two that are found in amino acids are circled in red, an amino group and a carboxyl group. You also have aldehydes, carbonyls, methyls, phosphates, and we'll come back to phosphates later when we talk about DNA. DNA has a critical phosphate group in it, and of course, a sulfate group. Amino acids and human health. So the 20 amino acids that are found in proteins, of them, our bodies can synthesize 10, half of them. The other 10 we must eat, get them from the food you eat. And so the essential amino acids, that is the ones you must get from food, are listed here. I won't go through them all, but simply say that complete amino acids contain all the essential amino acids, and complete proteins are found generally in animal products, eggs, milk, meat, fish, poultry. Incomplete proteins tend to be found in plant products. And so some examples are listed here. Rice, wheat, and oats have complete amino acid sets, but they're missing one, lysine. Beans, on the other hand, has lysine, but it's missing methionine and tryptophan. Peas are missing methionine, corn is missing lysine and tryptophan, and the list goes on and on. However, that doesn't mean that if you're a vegan or a vegetarian, that you're malnourished. Because in the case of, for example, rice and beans, if you had a diet of only rice and beans, you'd have complete proteins. Because beans supply the lysine missing in rice, and rice provides the methionine and tryptophan missing in beans. So anybody have questions about that? You see how it works. If you prefer to eat nothing but plant products, it's fairly easy to make sure you get all of the essential amino acids. All right, we'll go from proteins to carbohydrates. Carbohydrate means a hydrate or water of carbon. Carbohydrates are always composed of just 3 elements, carbon, hydrogen, and oxygen, and the ratio is always 1 to 2 to 1. So the empirical formula of a carbohydrate is CH2O. Any word, any name of a substance in biology that ends in ose, O-S-E, is a sugar. So here, for example, glucose, sucrose, ribose, they're all sugars. So remember that if I ask you to pick out of a list of names all those that are sugars, you look for all the oses.
Question. I
Yeah.
feel like this is probably obvious, but does all sugars end in ose? Like all of them?
As far as I know, I can't think of a sugar that doesn't, although there may be exceptions.
Yeah,
In
fair enough.
biology, in biology, there's almost always an exception. So
Yeah,
I can't say dogmatically, but in general, for your purposes, os means sugar.
gotcha. Thank you.
Monosaccharides. Mono means one, so these are single sugars. Glucose, for example, is a monosaccharide. You see its structural formula here on the right. Monosaccharides are highly soluble in water, good sources of energy. They're important as building blocks for molecules like amino acids or nucleic acids. And they're often part of macromolecules. So let's take a look at glucose. And one thing I want to point out here, the first thing in this illustration, is the numbering of the carbons. So glucose is a 6-membered ring. 5 of the members in the ring are carbons, and one, this blue atom here, is oxygen. So the number 1 carbon is the one adjacent to the oxygen. And then it goes around number 2, number 3, number 4, number 5, and then number 6 sticks out. It branches off of the cycle. So that's how carbons are numbered, and that's going to be important in a couple of minutes. Most carbohydrates, including glucose, can exist in either a cyclic form or a linear form. And on the left, you see a linear molecule of glucose. Same 6 carbons, but now linear instead of cyclic. In solution, about 97% of the molecules are cyclic, and only about 3% are linear. So glucose much prefers to be in a cyclic form. Isomers. Note this down. Remember what isomers are. They're molecules with the same chemical formula, but different structures. So here you see 2 molecules, 2 monomers. They both have the same formula, C6H12O6, but different structures. So glucose has a terminal aldehyde group, whereas fructose has an internal carbonyl. So it doesn't change the C's and H's and O's, it just changes the arrangement of them. And that can have a very significant functional difference. For example, I've indicated here that this change in configuration changes the taste. So if you taste glucose and then wash your mouth out and taste fructose, fructose will be sweeter. And you may have noticed sometimes when you buy groceries, a label might say contains high fructose corn syrup. Well, why is it high fructose corn syrup? It's because fructose is sweeter, and therefore the manufacturer needs to use less of it to get the same amount of sweetness. Disaccharides, 2 molecules joined together. Examples, maltose, lactose, and sucrose. And here is an example of maltose. Again, there's a dehydration reaction between 2 glucose molecules. You have 2 H's and an O that are eliminated as water over here. And that joins the number 4 carbon of one molecule to the number 1 carbon of the other molecule. And so this is called a 1,4 linkage. And we'll talk about that next, I think. So this is a 1,4 linkage that takes two glucoses and makes a maltose. Polysaccharides. These are long chains of monomers. And in the case of starch, glycogen, and cellulose, they're all composed entirely of glucose monomers. But they differ by the linkage type. And that's why I've been mentioning alpha in carbons 1 and 4 or 1 and 6. And I'll tell you the difference among these linkages in a bit. But first of all, starch, found in plants, is long, both branched and unbranched chains. The emphasis here is long. The linkages can be alpha 1,4 or alpha 1,6. Glycogen, found in animals, is the same thing except the chains are short and highly branched. Cellulose, found in plants, is very long chains of glucose, not branched at all. They're held together, the chains are held together by hydrogen bonds. And the linkages are beta 1,4. So let me explain what's the difference between alpha and beta and 1,4 versus 1,6. For the first illustration here on the upper left, you see two glucose molecules, an alpha glucose and a beta glucose. Well, what's the difference? You can see the difference in this hydroxyl here on carbon 1. If the hydroxyl is away from the oxygen atom, it is alpha. If it's on the same side as the oxygen atom, it's beta. So you see that here in a chain. At the top is a chain of glucose with alpha linkages. And at the bottom, the first linkage is beta. The connecting oxygen is on the side of the oxygen atom. Now, what about alpha and beta? So as I said, I pointed out the way that carbons are numbered. 1, 2, 3, 4. This is carbon 4. And on this other molecule, the critical carbon is number 1. So this is a 1,4 linkage, and it's alpha because the oxygen is away from the oxygen in the ring. So that's an alpha linkage. And in this bottom chain, you see two alpha 1,4 linkages. And this other arrow points to an alpha 1,6 linkage. And that's because the linkage is between carbon 1 of one glucose molecule and carbon 6 of another. And it's these 1,6 linkages that are usually associated with branches. So 1,4 linkages usually keep a continuous chain. A 1,6 linkage leads to a branch.
Professor, I have a question. This is Gabriela Galvez. My question is, all these saccharides and polysaccharides, what is the function then they have in our bodies? Because this is all about sugar, right?
Yeah, that's a good question. There are many different functions that depend on the type of linkages between molecules, and sugars is just one of them. Now it's really beyond the scope of this course to go into many different types of linkages. More than this, you should know that in the case of sugars, what are alpha linkages, beta linkages, and 1,4 and 1,6 linkages. But the physiological effect of those is complicated, and you can talk to a physiologist or you can look it up on the web, but it's not something that we want to discuss here.
Thank you, Professor.
Okay, this illustrates a point that I started to make earlier, and that's the difference between starch, glycogen, and cellulose. So starch is very long chains, highly branched. Glycogen is the same thing, except the chains are short and even more highly branched than in the case of starch. The branches, as I said earlier, the linkages in a chain tend to be alpha 1,4 or beta 1,4, but anyway, they're 1,4 linkages. Whereas when a branch occurs, it's almost always 1,6. It could be either alpha or beta 1,6. Now, cellulose is distinctive among the carbohydrates in that it has no branches. Cellulose is nothing but very long chains of glucose monomers that are bonded together by hydrogen bonds, not covalent bonds, but rather hydrogen bonds. And as I said, they're unbranched. The linkages are 1,4, but they tend to be beta. There can be alpha, but they're usually beta linkages in cellulose. And now we turn.
Professor,
Yes.
just a question. So you said they tend to be, but they're not, that's not necessarily that they are beta linkages?
In cellulose?
Yes.
Yeah, they can be either. They tend to be beta, but there can be alpha linkages in cellulose.
So does that matter as far as their effect of whatever they're in, or it doesn't matter?
Yeah, again, that's a good question. I don't know the answer to it.
Okay.
I suspect it matters, and I don't know what the difference is.
Oh, cool. Thank you.
Lipids contain mostly carbon and hydrogen linked together in nonpolar covalent bonds. Covalent, so they're very strong bonds. They're defined by their solubility properties. So they're soluble in organic solvents, nonpolar solvents. They're not soluble in water or in other polar solvents. Because they're mostly nonpolar, they are hydrophobic. Functions of lipids. Well, lipids, many of them are fats, and fats store energy. So one of the main functions of lipids is storage molecules, typically in the form of triglycerides, which we'll discuss in a minute. They're components of membranes, which is a critical function of life. They provide waterproof coverings around cells as well as insulation. Types of lipids: fatty acids, triglycerides. Now, among the triglycerides, there are fats and oils, and I'll tell you the difference in a little bit. But fats are solid. For example, bacon fat, lard, and butter, they're all solids. Oils are liquids, and oils come generally from plants: corn oil, coconut oil, olive oil. Another type of lipid is a phospholipid, and we're going to talk a lot about this because phospholipids form membranes. Steroids, such as vitamins, hormones, and cholesterol is a steroid, and then waxes, like beeswax or earwax.
Hey, Professor, can you go back to that one slide, to the slide before?
Yeah.
What does triglycerides mean?
What does triglycerides mean?
Yeah.
If you hang on for, I think, 1 or 2 slides, I'll show you.
Okay.
Okay, this is a hydrocarbon. I should say it's a hydrocarbon that is a fatty acid because it has a carboxyl group here at the left end. That's what makes it an acid. But you can see it's nothing but a long string of carbon atoms, and each one has a covalent bond with hydrogen atoms. So you remember that carbon, it's ubiquitous in biological molecules because it has 4 valence electrons. That means it can either pick up 4 or it can contribute 4 to bonds. And the 4 don't have to always be hydrogen atoms. They can be other types of group as well, which I'll show you in a minute. So here's an example of a saturated fatty acid. You can see that this long carbon chain, they're all single bonds. Carbon to carbon bonds are all single bonds. And there's a carboxyl at the end. This is a rigid molecule that it cannot bend or twist. Now this is an example of a mono unsaturated fatty acid. And it turns out to be a flexible molecule. It can either bend or twist because it has a double bond at some point between 2 carbon atoms. Again, there's a carboxyl at the end, making it an acid. It's mono unsaturated because it has 1 double bond. And here is a poly unsaturated fatty acid because it has 2 double bonds, or there could be more in a poly unsaturated fatty acid. And again, a rigid molecule like this tends to be solid, whereas a flexible molecule tends to be liquid. And polyunsaturated are even more liquidy than mono. Now you ask a question about what is a triglyceride. Well, this illustrates what it is and how it is formed. So you start out with a fatty acid, this long chain of carbons, which ends in a carboxyl, and glycerol. Glycerol is a 3-carbon molecule. It's an alcohol, and it has 3 hydroxyl on each of the 3 carbons. So again, a dehydration reaction is important here. So if a hydroxyl from the fatty acid makes a bond, loses a water molecule and makes a bond between this terminal C and the oxygen and glycerol, you now have the beginning of a fatty acid. It's a dehydration reaction. If you do that 3 times with glycerol, you now have a triglyceride, which is a fat molecule. And what you see here are 2 saturated molecules and 1 unsaturated one. And you can see that the unsaturated one is bent, and that has a profound effect on some of the functions of triglycerides and living cells. We'll talk about that. What you eat, you can eat foods high in saturated fatty acids, which probably not too good for you, or high in unsaturated fatty acids, and these are better. As I have said, the saturated ones tend to be solid. The unsaturated ones tend to be liquid. And here are some examples of foods that you should watch what you eat. Don't eat too much saturated fatty acid. Often labels on foods will indicate how much saturation there is in the fatty acids.
Also, sorry, but I found through a lot of foods that while they don't say they have saturated fats, I have an app where you can like scan it and it goes through it. So a lot of foods will say they don't have any, but they still have a little minuscule amount in them as well. So sometimes it's good to double check if you're particularly want to be healthful.
Yeah, well, thank you. That's good to know.
What's the name of that app?
It's called Food You Kate. I'll put it in the chat, but it's really cool. You basically like scan the barcode of anything, and if they have it in their database, it'll pop up, and they have like a team of nutritionists and scientists, and they go through every little ingredient and thing inside of there, kind of gives it a rating, and then it gives you alternative foods that you could get. Like instead of, you know, mac and cheese, you can get like Annie's organic mac and cheese and stuff, and it'll suggest different things for you. Yeah,
Nice.
it's pretty rad.
Thank you.
You're welcome.
Okay, yeah, thanks, Anthony. Appreciate that. But you make a even more general point, and that is most foods are not devoid of, if they say they're non something or other, they're not really non, but a reduced amount. A good example of that is decaffeinated coffee. It is decaffeinated, but it still has caffeine in it, just less than regular coffee does. Phospholipids. Now it gets interesting. Phospholipids are what's called amphipathic. Yeah, and I'll talk about that next. But that means, amphipathic means that they have a polar end and a nonpolar end. So the polar end here is to the left, the nonpolar end is to the right. The nonpolar end is composed of hydrocarbons, either saturated or unsaturated, nonpolar tails. They are hydrophobic. The other end of the molecule is called a polar head. It always has a phosphate group here. And then another group, which is also polar. It's usually some nitrogen containing group, but this can be variable. So don't worry too much about the variable one. Just know that there's always a phosphate group here, and that's why it's called a phospholipid. I want to emphasize amphipathic. I put a circle around it so you'll remember it. It's a molecule that has a hydrophilic or polar portion and a lipophilic, nonpolar portion. In other words, it either loves water or it loves fat. And what you see here in this illustration is an aqueous phase and a non-aqueous phase. And in the aqueous phase go all of the polar groups in an amphipathic molecule. So you can see each of these groups in the aqueous phase are charged. In the non-aqueous phase, they're all non-charged. They're non-polar. But remember the word amphipathic and remember what it means, because it's going to come up again and again. Phospholipids in water. This is how you build a membrane. So if you had phospholipids And you poured the phospholipids into a container that has water, which is polar, and oil, which is nonpolar. The phospholipid molecules would immediately orient themselves so that their nonpolar tails would be in the oil and their polar heads would be in the water. Now, this feature of phospholipids is the basis for biological membranes. And without this, without these amphipathic molecules, it would not be possible to make membranes, and without membranes, there could not be cells. Phospholipid bilayer. So on the left, you see an illustration of a phospholipid bilayer forming a membrane where the red tails are pointing in and the blue heads are pointing out. Outside the cell is aqueous on both sides. Inside the cell is nonpolar. It's highly lipid. And then these brown things are proteins that we'll talk about later. The proteins are what cause the cell to work. Can transport materials from one side of the cell to the other. It's very difficult for an ion, for example, to get through this membrane by itself. And here on the right, you have a space-filling model of the nonpolar tails attracting each other, making for the center of the membrane, and then the aqueous, or the polar heads in the aqueous environment that's both outside the cell and inside the cell. Steroids, another class of lipid. Whenever you see this structure, this chemical structure, 4-ring carbon unit, that defines a steroid. Now, steroids can have various side chains. You'll see that all of these steroids have the same basic structure, but testosterone differs from estrogen. They're both sexual hormones, and they both differ from cholesterol, which has a completely different side chain. And finally, we'll talk about waxes just a little bit. A wax is a fatty acid linked to an alcohol. And they are more hydrophobic even than our fats, completely insoluble in water. Why are they important in biology? Well, plant leaves are covered with wax, as are fruits. Birds' feathers are waxed, and you have wax in your ear canal. And this just shows you what I pointed out in the previous slide. Take an acid and an alcohol. The acid is characterized by this carboxyl, the alcohol by a hydroxyl. You have again a dehydration reaction, take out a water molecule, and it makes an ester bond. And so waxes are esters. They have these long non-polar chains joined by an ester bond, which itself is polar, but it's such a minor part of the molecule itself is highly non-polar. And I think that is all I have for that. Yes. So I need to stop sharing my screen and switch to a different slide set. it didn't work. More DNA and RNA. DNA is a molecule that makes up the genome of cells, but it's largely a library. And DNA is not very dynamic, whereas RNA is a workhorse. I'll talk about the various types of RNA and what each one does in the workings of a cell. Basically, it takes information out of DNA and makes it useful by constructing proteins from it. I want to tell you a little bit of history for a couple of reasons. One, I think it's interesting, and number two, it tells you something about a different way of doing science. We talked earlier about the scientific method and how scientists use this method for discovery. Well, here's a case where the method was not followed strictly. So, discovery of the structure of DNA in 1951, 3 people, James Watson, Francis Crick, and Rosalind Franklin, discovered the structure of DNA. And this was amazing, and it's had a profound effect on life in general. On the right here, you see a picture of Crick in later life. He eventually moved to the Salk Institute in La Jolla, and he died a couple of years ago. James Watson is still alive, and he was the director at the Cold Spring Harbor Lab for some years, but he no longer is, largely because of his political views. Now, I want to go back a little bit and tell you about why Watson and Crick and Franklin discovering the structure of DNA was such a monumental event. So in the 1940s, and prior to that, ever since the discovery of genes by Mendel, and we'll talk about Mendel's discovery of genes later, but nobody knew what they were. What's the chemical nature of a gene? And most people, most scientists thought that genes were proteins. And they had good reason to think that before 1950. And that's because proteins come in this amazing variety of forms. And in principle, the form of a protein could show the effects that we see shown by genes. So the hypothesis was that each heritable trait, the color of your eyes or the length of your fingers, each heritable trait is controlled by a specific protein. And since we now know that we have about 23,000 genes, it would only take about 23,000 proteins to account for every gene. Before the structure of DNA was known, it was thought to be a boring molecule, not really interesting at all because it was composed of only 4 variable parts, the so-called bases. And at the time, they were claimed to be present in identical quantities, and that is critical, and it turned out to be wrong. But no one prior to Watson and Crick imagined that genes are coded. But they had some hints. In 1943, a man named Oswald Avery showed that he could purify a substance from cells, and when he exposed avirulent bacteria with his substance, they became virulent. And the substance that he had purified was DNA. But you can see almost nobody believed Oswald Avery's experiment proved what genes were made of, because it was easy to say, okay, so he purified DNA, but it was contaminated with protein. So it was really a protein that had the effect. But then in 1950, a man named Erwin Chargaff showed that this conclusion was wrong, that the amount of A precisely equaled the amount of T, and the amount of G precisely equaled the amount of C. And there was no variation in this, no matter what form of organism, bacteria or mice or whatever you took the DNA from, A always equaled T and G always equaled C. Now this was highly reproducible, but no one knew what to make of it. No one knew what to make of these equivalencies. So to make a long story short, in 1951, Watson and Crick were working in the Cavendish lab at Cambridge. Rosalind Franklin was working at King's College in London. Rosalind Franklin was one of the few people in the world who was not only an expert crystallographer, she could actually crystallize pure DNA. And she did. And once she had crystals, and crystals, as you know, mean because it's crystallized, it's a pure substance. So there's no protein here. This is pure DNA. And when she's shown X-rays through her crystals, here's an example of what she saw. These bands. Now, Watson knew that regular substance-like crystals have X-ray diffraction patterns that are characteristic of the molecule. So when Franklin showed him this picture, he didn't say anything, but he immediately knew from this picture that DNA is helical. And that it has regular repeats. However, it didn't tell him whether there are 2 or 3 or 4 strands in this helical model. So instead of doing experiments, what did Watson do? He built models. And late in life, you see here, he's an old man, but he demonstrated how building models with cardboard cutouts revealed that the helix is double-stranded. And he did this by cutting out the shapes of the bases. T has one shape, A has another shape. And he could show that if you match them up together, he could demonstrate where the hydrogen bonding occurred. And he also had models of G's and C's. And by putting this together, just shuffling them around on a tabletop, he had this insight that it must be 2-stranded double helix. And I said earlier that they didn't take a typical scientific approach. Here we have our steps in experimentation. And Watson and Crick omitted this part. They may have had hypotheses, but they did not do investigations. They didn't do experiments where they had treatments and controls. Instead, they built, Watson in particular, built models with pieces of cardboard. So their approach is not typical of the way scientific discoveries are usually made, but it worked. And in science, whatever works is what you want. So the 4 types of nitrogenous base in DNA are adenine, thymine, guanine, and cytosine. And we come back to this picture, and I show it again because it is so important. DNA and RNA molecules are built from nucleotides made from just 3 main components: phosphate in the yellow, 5 carbon sugars in the blue, and a nitrogenous base, and it has to be one of these 5. If it's DNA, it's A, T, G, and C. If it's RNA, it's A, G, C, and U. Uracil is found only in RNA. And so this strand is an illustration of DNA. How do we know?
Because it only has A, T, D, and G. I mean, C and G.
Yeah, that's one way. It doesn't have any uracil. So that says immediately, there's another outstanding feature here that says it's DNA, not RNA. RNA is ribose nucleic acid, and DNA is deoxyribose. The blue is the ribose molecule. So what is there about that that makes it DNA?
Well, that DNA has 2. What is that?
This is DNA, this is deoxy. So I'll tell you, it's carbon number 2 here. It's a hydrogen. If this were RNA, it would be a hydroxyl, OH. And so you'll see that whenever you see the structure of an RNA molecule. Here we have
double-stranded. Okay.
The structure of a double helix. The ribbons are the sugar phosphate backbones, and you see the bases are indicated by G's and C's and A's and T's, and they are joined by what? What did Watson, when he put his models together, what was he looking for?
Hydrogen. I mean, was that hydrogen?
What is these bright lines are hydrogen bonds.
Right.
The hydrogen bonds are between the bases. So all of these bonds that are holding these chains together are hydrogen bonds, but always G is always bonded to C, A is always bonded to T.
Okay.
Now, I want to make a point with this, that first of all, to repeat, it's always T and A, or A and T, it's always G and C. But you'll see that T and A are joined by blue bars. Those are 2 hydrogen bonds between A and T. With G and C, there are 3 hydrogen bonds. So remember that. I think it'll show up in your worksheet. But between G's and C's and A's and T's, there's a difference in the number of hydrogen bonds. Now, what is a gene? It is nothing but a section of DNA that codes for another molecule. So here's a section of DNA. If it's the right sequence, it will encode another molecule. If it's not the right sequence, it will encode nothing. It's considered junk. The sequence of nucleotides for each gene is unique for that gene. The nucleotides are always read in groups of 3 letters. We'll talk about that, I think, the next time. The human genome contains about actually 23,000 protein coding genes, and that makes up less than 2% of the total genome that each of us has in each of our cells. So that immediately begs the question, if it only takes 2% of our DNA to account for all of our proteins, all of our features, all of our characteristics, what about the other 98%? What does it do? Well, in a nutshell, much of it is unknown, but the more geneticists study it, the more functions they're finding. And by and large, most of the functions of non-coding DNA, non-coding means they don't code for proteins, most of these are regulatory. They determine when a gene is on and when a gene is off, and if it is on, how fast is it running? So these are transcription factors, could be introns or pseudogenes or enhancers or silencers. They're all kind of non-coding sequences that control what the genes do. And we'll talk about more of that later. And this comes back, remember you were asked at one point to think about how we have the same 23,000 genes in each of our cells. So how do we get differentiation into different cell types? And this is how it's done, because of these non-coding sequences regulating when genes are on or off and how fast they run. Genes are located on chromosomes, and if you're a cytologist, you could make a karyogram, which is a karyogram is a picture of chromosomes. Now this is a picture of a human female chromosome set. Humans have a total of 46 chromosomes, 22 pairs of what are called autosomes. These are that function in the body, and 1 pair of sex chromosomes, which function only in the sex organs. Now, females have 2 of these sex chromosomes, an X and an X. Males have 1 X and the other chromosome is called Y. Now this introduces us to the CFTR gene, and we're going to talk about that more soon in subsequent sessions. And I'll get on to describing your project. But the CFTR gene is responsible for cystic fibrosis. Its wild type version is called the cystic fibrosis transmembrane conductance regulator. And what does it do? It determines the movement of chloride ions across membranes. Remember I said earlier that membranes are highly impermeable to most things, and to get across, they need help. And the CFTR gene encodes the protein that helps chloride ions get across the membrane. And what you're going to be working with are mutants of this gene, and you're going to ask the question, okay, what's wrong with my mutant? Why is it not transporting chloride ions? And the consequences of the failure to transport chloride ions are quite severe, and they result in this disease known as cystic fibrosis. So this showed up on your worksheet, and those of you who have tried to do the worksheet already came up against a blank here because you were asked, what does this code mean? Well, it indicates the chromosomal location of a gene. And so the code here is 7q31.2. 7 means it's the 7th chromosome. So as you know, chromosomes are numbered from 1 to 23. This is chromosome 7. The Q means the long arm. So every chromosome has a centromere. The centromere divides the chromosome unequally to 2 arms. The long arm is called Q, and the short arm is called P for petite. Then, so Q means long arm, 3 means region 3. So you see this boxed region here at the bottom of the chromosome is region 3. Band 1 is the darker little box here, narrows it down more. And subband number 2 narrows it down to just this one band where the CFTR gene is located. So that is, that's the answer to the question on your worksheet. The CFTR gene is very large. You can, we don't have time to do it now, but you can look up this website and see just how large it is. It's huge. Over 150 million base pairs. It has 26 introns and 27 exons. We'll talk about what are introns and exons later.
Okay, Professor, I have a question. Is that this is the answer for what is the meaning of 4p to 1.3?
Yes.
Okay.
Yeah. And and I apologize for getting ahead of myself with that, but I assigned the worksheet before I had covered the material in class. So yes, the code you see on your worksheet, it's not this code, but it's something different. But now you know how to interpret it. It's the chromosome number, the arm, and then the bands and subbands where a gene is located. Okay, that is the end. And we are just out of time. So I'm glad we made it. Now you have all the information that you need for the worksheet and for quiz 3, which will be on Wednesday. And
But you did say we will have class on Wednesday. We will have class.
we will have class on Wednesday. It's just that it will be 60 minutes instead of 75.
Okay.
But on next Monday, you will not have class because you'll be required to take the full class period to do test 1. Okay, session is over.